For part 2, we're continuing on with the central dogma of molecular biology, kind of showing you the flow of the genetic information. And we're going to focus on two main processes, transcription and translation. And in order to do that, we also need to talk about RNA. And there's 3 different types of RNA that you're going to need to know. Those 3 types are messenger RNA, which we abbreviate mRNA, ribosomal RNA, or rRNA, and transfer RNA, or tRNA. Messenger RNA carries the coded information, or the message, for making proteins from DNA to the ribosome. So it basically copies the code from DNA and then brings it to a ribosome, because remember, ribosomes are where proteins are made. Ribosomal RNA is pretty easy. It just forms the ribosomes. Okay? So it helps make up the ribosomes, which play a role in making of proteins. Transfer RNA transports amino acids, so that's its job. It's going to transfer that amino acid to the ribosome, where those can then be assembled into proteins. So the order of the amino acids is what helps make the protein. So you'll need to know the jobs of each of these 3 types of RNA. All right, transcription. This is when DNA is transcribed to make RNA, or another way of putting it, the DNA code is copied onto RNA. Specifically, messenger RNA is one that carries that code. Okay? But we need to know where to start transcription, because we're mainly interested in copying genes. In other words, those segments of DNA that code for protein. It would be a waste of time to copy every single bit of the DNA and carry that to a ribosome when only certain chunks of it, only certain pieces of the DNA actually code for proteins. So we need to know where to start. And so there is a specific sequence on the DNA called the promoter region. And this is going to signal where transcription begins, because it's going to tell RNA polymerase where to bind to the DNA. So it signals the beginning of a gene. Okay. Transcription proceeds in a 5' to 3' direction. Okay, because that's the only direction the RNA polymerase can go. And then transcription is going to stop when you get a terminator sequence. So when it reaches that final kind of stop sequence or termination sequence, that signals the end of the gene that needs to be copied. Okay, that way we're not wastefully copying parts of the DNA that really aren't coding for anything. We just want to get that gene that codes for that specific trait. RNA polymerase is going to bind to that promoter region on the DNA. Remember that there will still be gyrases that are going to relax the supercoiling. There will still be helicase enzyme that's still going to unwind the DNA and break those hydrogen bonds to separate it, so that the RNA polymerase can then bind to that promoter region. And it only moves in a 5' to 3' direction. And its job, very similar to DNA polymerase, is going to be adding nucleotides. And these are going to be RNA nucleotides that are going to be complementary to the DNA nucleotides. It's just that any place where you normally would have put a T in DNA, now uracil, the U, replaces the thymine. So let's say this was DNA and this was RNA. We had T, A, C, and G. On the RNA, you would have A, because A would still bond with T, but here you'd have a U, because U will not bond with A. You still have G, We still have C. So we still have A, G, and C, but now U replaces the thymine. The sugars are different in RNA as well. It has a ribose sugar, so RNA is called ribonucleic acid. Okay, so RNA, like that. It is single-stranded as well. So it's not, it's still twisted into a helix, but it's not a double helix like DNA was because it only has a single strand. And transcription is going to stop when the RNA polymerase reaches the terminator region. Okay, when it reaches that, that's going to stop transcription. Then the RNA polymerase and messenger RNA get released, and then the DNA winds back up again until more of it needs to be copied at a future date. And then the messenger RNA is going to carry that message to the ribosome so that translation can take place. So here it kind of shows you. Here's the promoter, and that's just going to signal where the gene begins. And so then the RNA polymerase knows where to hook on, and then it goes down the line and starts putting the correct bases. A with T, here's the U bonding with the A, and so on. It goes all the way down the line. It will continue until it reaches the termination sequence, and that just shows you that that's the end of the gene. And then everything breaks away. Here's the messenger RNA that's now going to travel and go to the ribosome, and here's the RNA polymerase that's going to break away as well. Now, we need to talk about what do we mean by the genetic code. The genetic code is just the way that we read the bases in order to help use that information that's in the bases to help make a protein. So we have some terms for you to know. Codon is just a group of 3 nucleotides on messenger RNA that code for an amino acid. And there can be 2 different types of codons. The sense codons are the ones that code for amino acids, and the non-sense codons code for stop codons. Okay, they do not code for amino acids. They are what we call the stop codons, and they will just code for a stop sequence. Anticodons are a group of 3 bases on transfer RNA. Okay, a codon is found on messenger RNA. The anticodons are those 3 bases that are complementary to the messenger RNA codon. So they're going to be the exact opposite of the messenger RNA codons. And this is what allows, remember the job of the transfer RNA is to carry an amino acid to the ribosome so that they can be placed in the correct order to make a protein. Well, the only way transfer RNA knows where to go to the appropriate spot on the messenger RNA is that it's going to match its anticodon with the codon on messenger RNA. Now, we do talk about what's called the degeneracy of the genetic code. Degeneracy means that several codons can code for the same amino acid. And that really is a way of protecting us against mutations, because if there's a change in that codon, then maybe it's still going to code for the exact same thing it normally would have. So then it's like a silent mutation. So it protects us by having the degeneracy of the genetic code. So I'm going to show you what I mean by that. Here's 2 different ways of showing the genetic code. This square box here, this was the old way, and this is the new way, which I love a lot better. I had to learn on the old box thing. I think that circle is so much easier. How we read this, let's say I wanted to find out what amino acid, these on the outside here, these are amino acids. Amino acids. Okay, so in this very outer ring here, that's what all of those are. Okay, so we read these starting with a middle and working our way out. So let's say I wanted to find A, This is my codon on the messenger RNA. I would start first here in the center circle. Here's my A right here. That means I have to stay within this piece of the pie for the rest of the thing. I now move out to my next circle, and it's a C, so it'd be this one right here, because that's the next circle, okay? That means now I stay in this piece of the pie, and then the third circle is right here, and here's the C. Actually, all of these code for the same thing. So ACC codes for threonine, all right? Because of the degeneracy of the genetic code, if this got changed, instead of ACC, it was ACG. ACG still codes for threonine. So see, even if I would have that mutation, I'm protected because of the degeneracy of the genetic code. The fact that more than one codon codes for the same amino acid. Okay? So once we have that codon messenger RNA, we're going to use those codons and the genetic code to translate what that means. We're going to look at those codons and start to determine what amino acid sequence we have. And by using that amino acid sequence and putting those amino acids together, we are going to make a protein. So first of all, the messenger RNA and the ribosomal subunits get assembled. So your rRNA is going to help out with that. The transfer RNA is going to bring the first amino acid to pair with the start codon on messenger RNA. Then you'll have another transfer RNA carrying a second amino acid, and that attaches to the next codon. Then we have what's called a peptide bond, which is going to join the two adjacent amino acids. So here we have a large subunit right here and your small subunit. That messenger RNA is going to hook on right between them. And this is what we call right here our start codon. Okay? If you look that up on your genetic code chart, AUG, so AUG codes for methionine. So that's always going to be the first amino acid that's going to come along and be put there. And you can see right here it's abbreviated MET. So here's your transfer RNA. It's carrying the amino acid, but it has to know where to bring it. So it's got this anticodon here, UAC, which is complementary to AUG. So once we have that complex formed together, you'll notice that there's two big spots here. Okay. The first site here is called the P site. Here's a spot for that very first transfer RNA that brought its, that's matching up its anticodon with the codon, but it's carrying that amino acid on the end. The next one will come along and fit in. UUA is the codon. AAU is complementary. This is the anticodon. So that transfer RNA is going to come along. It's carrying this amino acid on it, and it's going to bring it into that second spot there. Once you have two side by side in the ribosomal complex, now you'll have that peptide bond forming between these two. Once we form these, the ribosome shifts down a spot. Okay, the ribosome moves along the messenger RNA, exposing a new codon where another transfer RNA can bring another amino acid. That initial transfer RNA gets released to pick up more amino acids, and it's going to continue down messenger RNA until it reaches a stop codon. Once that stop codon is reached, the ribosomal complex falls apart, tRNA gets released, and then that polypeptide is released to form a protein. Okay? So, the polypeptide is released to form a protein. So here you can see we release this. This is going to move this one off, and it's going to detach because we've already bonded these two together. Notice it's already released its its amino acid. There's no longer an amino acid here. Okay, this is going to shift down. When it shifts down, it leaves a spot. These two will get bonded together. Then it's going to shift down another spot right here to leave another area. There it goes. And as you can see, you've got this growing polypeptide chain with all these amino acids joined together by peptide bonds. And then the old tRNAs just get released and they go out in the cytoplasm and go get more. It just continues and continues and continues until you reach a stop codon. Okay, UGA codes for a stop codon, which signals the end. And that means everything kind of breaks apart, and here you've got your new protein. And it's going to start folding and bending and all that to make the shape that it needs to be the protein. Now, the big difference between prokaryotes and eukaryotes is that transcription occurs in the cytoplasm. So transcription's in the cytoplasm. Translation occurs in the ribosome. Okay? With eukaryotes, transcription occurs in the nucleus because eukaryotes have a nucleus. They also have to, after transcription, the RNA has to undergo processing before leaving the nucleus. So the exons are the segments of DNA that code for proteins and are expressed, thus the exons. Introns are intervening segments of DNA do not code protein. So that's why we want to get rid of them. The only part we want of the DNA is the part that codes for proteins. And so we kind of splice out or cut out those introns and we squeeze all the exons together. Translation still occurs in the ribosome, just like it does up here for the prokaryotes. So remember, prokaryotes do not have a nucleus. They just have that nucleoid region that's found in the cytoplasm. So that's where translation would have to occur. This is just showing how in eukaryotes we have to take out all these introns. Those have to be cut out. And then you can splice together your exons. Okay?